Good morning, everyone checking the mic, and thank you to the Alliance for Regenerative Medicine for the opportunity to present. Today I'm going to present Splice Bio. My name is Gerard. I'm Chief Business Officer at the company. We're a genetic medicines company developing a novel gene therapy modality called protein splicing. Now, a bit of a summary or overview for your reference. So we have a lead program in Stargardt disease undergoing GLP IND enabling studies. We've developed a pipeline based on this platform of several programs with a focus in ophthalmology and neurology. We raised a $57 million Series A in 2022 and incorporated recently Anis Girac as our Chief Medical Officer. And we announced October last year, 2023, a partnership with Spark and Roche around one of the programs that we have in the retina. Here, there is a slide on the team. The company was founded by Mikael, Sylvia, and Professor Tom Muir, chairman of chemistry at Princeton. That's where the technology is coming from. And the round in 2022 was co-led by UC Ventures and Isios, and we had the participation of NEA in the States, Novartis, Gilde, and Asavis. And the nature of the problem we're trying to address, you'll mostly be familiar with it, right? It's the fact that we've had several approvals based on AAV vectors, but they all have a fundamental physical limitation, which is that they can only package 4.7 kilobases of DNA, right? So any genes that you like to deliver that exceed that packaging capacity, you cannot deliver them. Now, people have been trying to address this problem via different approaches. We're aware of alternative vectors, the mini genes that have resulted in some approvals recently, as well as the dual AAV approaches. Now, the issue with the dual AAV approaches is that they have relied mostly on recombination at the genetic level, right? Be it DNA or RNA, which we know is not that efficient, especially in terminally differentiated cells, which are the type of cells that you actually want to target with AAV vectors. So what is it that we're doing, right? And that's where protein splicing, meaning splicing at the protein level, comes in handy. So when we are faced with a gene that is too large to be delivered by a single AAV, we split that gene in half. We package each half of that transgene in two separate AAV vectors, and we append to that transgene the sequence encoding for small protein domains, quite obscure and quite mysterious, because usually people have never heard of those protein domains called inteins. And what inteins do is catalyze a natural process called protein splicing that results in the formation of full-length native protein. The way it works is you have expression, transcription and expression as usual with an AAV vector, and it is at the protein level that these two intein domains will recognize one another. They form a large protein-protein interaction that reconstitutes the full-length active catalytic site of the intein domain, and that triggers the protein splicing reaction whereby the inteins ligate the two fragments that had been expressed separately into the full-length native form of the protein, and inteins are self-excised from the mature protein, so the process is entirely scarless and traceless, and you can effectively reconstitute the functional version of the native protein that you could not deliver in the first place because it was too large, right? Now, inteins exist in nature, and they were discovered by our co-founders and others in the late 90s, so a legit comment is, is it that you are smarter than anyone and you figured this out when these guys have been around for 25 plus years? And the thing is that natural inteins are not that great when you're thinking of human therapeutic applications, right? They are fantastic as tool reagents, but they have numerous limitations that limit their potential application in human therapeutics. One of those being that you cannot simply take a random gene or target and split it right where you like and hope that the inteins will work. Because inteins do have quite strict sequence requirements. What that means is that you need to find usually 3, 4, 5 amino acids in what we call the split junction. And that's the -1, -2, -3, +1, 2, 3 residues in your target protein. And you need to find those residues for those inteins to be inserted there and for those inteins, natural inteins, to work. If those residues are not in your protein of interest, you'll not be able to split and reconstitute a target protein using protein splicing. So what we did at Splice Bio, and what our co-founders did, is realize that this was a significant limitation for, especially for the gene therapy space, right, where ideally you like to reconstitute the native form of the protein. And what we did was, based on this know-how of 25 plus years about inteins, is design and engineer an engineered intein, a consensus sequence that could get rid of all those sequence requirements. And now we can essentially plug and play these inteins across targets without this sequence limitation. And we've proven that. So as I said, inteins exist in nature, all domains of life, but only in unicellular organisms. They are autoprocessing domains, and that's one of the properties I like the most about inteins, is that they do not need any additional cofactor or enzyme or cellular machinery, for that matter, for them to be able to perform their function. Everything they need to perform the protein splicing reaction is encoded in their primary sequence. They're extremely versatile and relatively small as well. And so beyond the sequence requirements, the engineered split inteins that we've developed express excellently, and the splicing rate is 20 seconds, meaning in 20 seconds you have 50% of your splice product formed, which is extremely fast, right? And so this technology, we've been working on it for several years, and it's built on 25 years of pioneering research in protein chemistry and protein engineering, and we have validated it already in more than 20 targets. So essentially, any target we've intended to split and reconstitute with our technology, we've been able to. We have validated it in vivo across organs, retina, brain, inner ear, and muscle is what we've disclosed, and across species. So we have robust data showing that the technology behaves robustly and consistently in rodents, pigs, and obviously non-human primates, which is the most relevant. And this is how our pipeline looks like. So we have a lead program in Stargardt disease aimed at reconstituting the full-length version of the ABCA4 gene. It's in IND-enabling studies. We have 2 more programs in the retina undisclosed. The 3rd program in the retina has been partnered with Spark in a $260 million deal that we announced in 2023. And the 4th program is a CNS program that we are pursuing. Now, regarding the lead program, Stargardt disease is caused by loss of function mutations in the ABCA4 gene. ABCA4 protein is a transporter of certain metabolites to preserve metabolites that are the result of the regular visual cycle in the retina. The lack of ABCA4 results in the accumulation of these toxic metabolites and the death of photoreceptors and blindness. This is a representation of how Stargardt patients see. And this is how the drug product looks like, right? So we have 2 AAV vectors. Each of the transgenes is flanked by the promoter and the poly-A, as well as the ITRs. And we're delivering each of the halves of the ABCA4 gene. This is some proof of concept data in the mouse model of the disease. It recapitulates the biochemistry of the disease, but mice do not go blind, so you cannot assess clinical function. But what you can see on the left-hand side is the levels of expression that we can achieve with our SB07 gene therapy in dose 1 and dose 2, right? That's comparable to the endogenous levels of ABCA4 expression that you see in wild-type mice. We have some immunohistochemistry imaging data demonstrating that the protein localizes where it's supposed to localize. And we have also performed studies where you can monitor how the progression of that, how those toxic metabolites accumulate in the model, and how treating with our drug essentially blunts the accumulation of these toxic metabolites, indicating that the protein is functional and that we have a pharmacological effect. Now, the mouse model is quite limited. The retina is very small, so it's quite tricky in terms of doing those administrations. So we wanted to also confirm the pharmacological effect of SBO7 in a different model, the rat model, which is a bit more handy because it's a larger retina, and we essentially see the same data, right? On the right-hand side, what you can see is that we can blunt the accumulation of these toxic metabolites, in this case A2E, that accumulate in the model of Stargardt disease. Most importantly, we've done extensive work in pigs and in non-human primates. I'm not sure the lighting will favor being able to look at the images, so maybe I will transition to the non-human primate data. It's a bit of the same. Here, since we're using codon-optimized sequences, we can discriminate between the endogenous ABCA4 RNA and the RNA that we deliver with our therapy. And on the left-hand side, what you can see is the endogenous levels of RNA expression of ABCA4 in wild-type macaques. And on the right-hand side, you can see the signal that we can achieve with our SBO7 therapy in terms of RNA expression of each of the fragments, right? And at the mid-dose, you can see that we are already saturating the system, and as the pathologist report indicates, we have levels of RNA expression that are comparable to the wild-type macaques. Most importantly, in the macaques and in humans, one of the questions is whether a gene therapy can transduce not only rods, but also cones, right? Which in Stargardt are particularly relevant because they are the ones responsible for visual acuity and central vision. And what we can see in this image is that the RNA expression is also strong in the foveal area where you mostly have cones, mostly non-human primates and humans have 95+% of cone expression, and all this pink signal indicates that the RNA of ABCA4 that we're delivering is expressed as it should in the outer nuclear layer of photoreceptors. We also had some data showing protein expression in macaques. And to summarize, so the program is now undergoing IND enabling studies, and we intend to be in the clinic next year. And regarding the clinic, we recently announced the incorporation of a clinical team, including Anis Girak as Chief Medical Officer, that had been the Chief Medical Officer of ProQR and Nightstar before. And this is how the pipeline is looking like. So the lead program, SBO7 IND-enabling studies targeting the clinic phase 1/2 trial in 2025. We've had a bunch of interactions with regulatory agencies that have validated our path to the clinic and registration. Second program is a bit further behind, and the third and fourth programs are at research/proof of concept stage. And we're essentially raising Series B now with a goal of funding the lead program throughout a robust clinical proof of concept and initiation of a phase 3 trial, and the second program throughout a phase 1 clinical proof of concept trial, as well as continuing to pursue the pipeline that we've built. And yeah, that's pretty much it. We have a minute for questions if anybody would like to. Yeah, so they're relatively small, right? So the intein N fragment is roughly 100 amino acids. The intein C fragment is 35 amino acids. We haven't seen that in all the studies that we have conducted. We've done also some bioinformatic analysis to understand, to predict the presence of T cell epitopes, and we haven't found any. The AB capsid itself is what drives the immune response